Our final speaker is John Rex. John is Chief Medical Officer at F2G. That's an antifungal biotech. And I'm wondering, John is going to be by Zoom, correct?
Sound check okay?
Sounds good, John.
All right. Well, so thanks to the program committee for inviting me. This has been a great day. I have enjoyed listening all day long. And I'm going to talk about a developer's perspective on new drugs for COXY. And let me just tell you the punchline right now. It's a lot harder than I thought it was ever going to be to go into this area. My coordinates are shown on the slide. Drop me a note if you want a copy of it. So scope. Hart, I'm an ID doc who wants new antibacterials and antifungals. And my comments come from an experience, an academic from inside of regulated industry, all phases of development, antibacterials and antifungals. I'm going to focus on new therapies. You just heard about the limitations of current therapies. I think progress is going to take new. Vaccines would be even better. We'll hear about that tomorrow. And at times I will broaden the discussion to include invasive fungal infections in general. Sometimes that's helpful. I do work at a company that has a novel antifungal in development. I'm not going to talk about that, but I will talk about a PRO that we're developing in COXY. So some key regulatory principles you need to know. And this is your homework. FDA had a wonderful public workshop in 2020. All the materials are online and there's a published manuscript. You need to read this if you want to think about developing new drugs. I'm going to pick out a few things from their presentation. First, the drugs for treatment, as GR just said, two things that are approved, neither one of which we think of as a good drug anymore. So there's not actually a standard of care FDA-approved drug. There haven't been anything new for 20 years, according to the FDA and their slides. And that leads to the question of what comparator would you use if you were going to do a study? You're going to have to use a non-approved agent, but their FDA has flexibility for that. Regulatory pathways are worth spending a second on, and it's helpful to realize that there are two fundamental pathways. One's called traditional approval that is based on a classical endpoint about how somebody feels, functions, or survives. And then there's the thing called accelerated approval, where you're looking at something like HIV viral load, and you know that the disease won't occur for another 20 years, but you don't want to wait 20 years to find out if the drug works. And so you're actually going to use an HIV, you can use the HIV viral load as a marker. And that's what's called accelerated approval. So you're not waiting for 20 years to find out what happens to the patient. And on top of that, or parallel to it, there's an idea that called LPAD that I'm going to spend a slide on here in a minute. All these pathways require what's called substantial evidence of efficacy based on adequate and well-controlled clinical investigations. And that is a constant across the entire group. So when we look at endpoints, I talked about field functions survived, and I talked about surrogate endpoints. Just be sure this is really clear. We've got 5 possible endpoints here for drug approval: all-cause mortality, improvement in symptoms of pneumonia, drop in HIV viral load, conversion of blood cultures to negative in endocarditis, fall in the serum comp fixed titer from 1 to 16 to 1 to 2. Which one would be valid? Well, it's only the first 3. The first 2 are classical clinical endpoints. That's something grounded in how you feel, function, or survive. And the third, I talked about a second ago, HIV viral load has a really a one-to-one relationship to outcome of that viral infection. The other 2, while they're important, I want the blood cultures to become negative. I want the comp fix to fall. They do not have that tight linkage the way HIV viral load does. And so unless you did a lot more work to confirm their validity, you wouldn't be able to use them as an endpoint. And that's because endpoints need to be what's called well-defined and reliable, something that everybody can agree on, that everybody can measure over and over and over. And here you get into the issue that with clinical endpoints for COXY, it's really difficult to define even 1 or 2 symptoms, other than maybe fatigue, because there's such a wide variety of presentations. No single symptom is ever going to capture everything. And composite endpoints count the number of hot joints. Count the add up 3 or 4 symptoms. Composite endpoints are tricky because it's hard to weight the various elements inside the composite. The idea of a biomarker, the idea of a surrogate, I think I've said this now a couple of times. It's not that it's disallowed, it's just that you've got to prove that it actually has meaning. So there is this pathway called LPAD that was created in 2016, and if you're going to work in this area, you do need to know about this. And the first thing you need to know is it's misspelled. It gets abbreviated as LPAD, but actually it should be LPAAAD. Sing it out in Italian. LPAD, limited population antibacterial and antifungal drug. And the idea here is that we're working in an area where we have a serious or life-threatening infection, and there's a limited, well-defined, clinically relevant population that has an unmet need. And in that space, FDA is able to approve for that limited population, even though there may not be enough data to use it in a broader population. And that's the idea. You're approving just for that well-defined limited group. And FDA would then take into account the severity, rarity, or prevalence of the infection. Not an easier pathway, not a way to fix a failed program. Still got to do substantial evidence of effectiveness. You got to have data on how to put it in the label to tell you how to safely use the drug. But it's a valuable support for rare infections. 2 drugs approved to date with LPAD, inhaled amikacin for M. avium, pertamine for XDR-TB. So some preclinical issues. This is general stuff about all drugs, but there are some specific twists for Coxsackie. So the big question you start off with, is it a drug? And I always think of the cartoon out of XKCD, where when you see a claim that a common drug or vitamin kills cancer cells in a petri dish, keep in mind, so does a handgun. And so do steam, fire, and bleach. They're all very effective antimicrobials. It's the selective killing that's the subtle art. Here I guide you to the AAC instructions to authors and read on the AMR.Solutions website the 3-part series on halicin, a compound that was discovered by a machine-based technology, but it really turned out to not be an antimicrobial. Look at some other things. Making the drug, the land of what's called CMC, chemistry, manufacturing, and controls. How does your thing turn into a tablet? There's a lot of work that goes into creating that pill in that bottle. And I just want to summarize this briefly by saying it's a science unto itself. And you really need to get an expert on your team. There was a great boot camp in 2017 that we sponsored at one of the workshops. Go and look up the boot camp off of the Carbex, the GARP, or the AMR.Solutions website. PK, ADME, and PD. What dose? ADME, absorption, distribution, metabolism, and excretion, in case you don't know that acronym. Where does it go? Right body compartment? How many doses a day? Is it oral? For Coxsackie, it probably has to be oral. I think IV would be nice to have. Drug-drug interactions, stuff, just general usability of the compound. And then there's pharmacodynamics. What concentration do you need? So what dose are you going to put into human beings to get that concentration? Strong PK/PD is a key support for smaller programs. Deciding on a target exposure is a subtle art. There's a paper, Hope 2016, that lays out some ideas about how to do this for antifungals. Animal models for Coxsackie exist, but they require specialized facilities. Finally, is it safe? A lot of ways that you approach safety preclinically. There's some in vitro assays you do. But with respect to Coxsackie, it's important to know that before you can go into man, you're going to have to do in vivo supertherapeutic exposures of your new agent and its metabolites in 2 animal species for a period that's long enough to let you treat the people. And the rules about safety coverage and just dosing in man is you get 1-to-1 coverage for the first 90 days. You do safety study for 28 days, you can dose in human beings for 28 days. Do safety for 90, you can dose for 90. If you want to be able to dose for an indefinite period, you've got to do 180 to 270 days of preclinical studies. And since we're often going to be treating Coxsackie for a long time, means you're going to have to do that work. And that work takes a long time and takes a lot of drug. And you've also got to be assured that you test all the metabolites at the right exposure in those animals. This is a big block of work that you have to have done before you can get serious about exploring a drug for Coxsackie. All right, so with that as background, let's now talk about trial design. And when I was asked to give this talk, the real focus, the real request was, what kind of trials do you think you could possibly do? And so I spent a little time chewing on a way to display this. And so let's do a thought experiment. You've got a candidate compound, oral. You've got your phase 1 data, so you know some PK and you know the dose that'll give you your target exposure, and you've got safety talks out to day 90. If the spectrum permits study of another fungus, I will tell you, go do that instead, please. If nothing else, you might do phase 2 dose finding with another fungus, because as you're going to discover, doing this in cocci is hard. And at some point, let's say you finally decide you do want to do a controlled study in cocci. And let's start with the idea of the goal of a randomized controlled trial, sort of the classic perfect study. We've got a lot of things we need to consider. Think about trial populations. We've had several talks today where different manifestations of cocci were discussed. Let's start off with primary uncomplicated pneumonia. I think the difficult things about this are that it's hard to diagnose because the serum markers develop slowly, and the presentation is entangled with CAP, community-acquired bacterial pneumonia. And I think the FLEET study showed us how hard it is to separate all this out. And I just think that finding these patients will be hard. And it's also, I think, going to be harder to declare that you meet the criteria for LPAD. As I say, I think LPAD's helpful. Your compound's going to need to be pretty darn clean because you're going to be going into a general population. This just your tolerance for safety signals will be lower. Now, I will say that you might be able to improve on time to improvement. The observational data suggests that azoles don't do very much. And in parallel with that, it's actually kind of low medical risk if the compound doesn't perform. The azoles don't do anything either. So if your compound doesn't do much, it's kind of like nothing lost. But it looks like you might be able to make some progress with that. Overall, this one for me is tough, and I'm going to put a big red X on it. Chronic fibronodular fibrocavitary, that distinct but uncommon subset of disease in the lung. I think this is a distinct subset, and it's a pretty significant disease for people who have it. I think the downside would be, is it treating that form generalized to other forms of cocci? Hard to say. Azoles do have a little bit of an effect, but the therapy seems to need to be long, and there's a high recurrence rate, so maybe you could beat that with a new drug. And I'll say that I think the medical risk of a new drug not working terribly well isn't all that high in this setting. I think the big difficulty here is that this would be a long, hard trial. The patients aren't very common, and if you want to show a reduction in rate of recurrence, you're going to have to follow people for a couple of years. That's a tough study. So, big X. Let's get into the more complicated forms, and let's start with non-CNS and include in that progressive pulmonary. One advantage of this is it's a little more common than the next version we're going to talk about. I think you can make an argument about this, but I think the medical risk if the new agent doesn't perform is kind of in the medium range. You could probably salvage somebody with current therapies. I would say that I think bone's a little harder to treat. I might have studied it separately. Pro-con around what kind of design you'd pursue here. Azoles have a little bit of an effect, but responses take a long time. Recurrence is the problem. I think you might be able to improve on that. I think there's probably good potential in here for LPAD-based approval. So when I think about this one, I think, yeah, you might be able to do that. I mean, it's not going to be easy, but you might be able to do it. The last one would be CNS disease, less common than non-CNS. But I think the big difficulty here is this is high-stakes poker, serious disease. Do not get the dose wrong. The upside of this is it's the most challenging form. If the new agent works, that's very impressive. And could you do a superiority design? Well, maybe. Fluconazole or the azoles in general have some effect, but response does take months. Not clear how or when you can ever stop. So there might be room to improve here. I still think it's a tough study. And I think it probably does have good potential for LPAD-based approval. So that one, for me, gets another definite maybe. That would be a place I think you might be able to do a study. Think about what we're going to measure in our study. Got 3 endpoints to think about. First is a traditional endpoint, and here I'm going to point at the EORTC MSG scoring system, which is based on the triad of clinical, radiological, and mycological response, and success requires you to get better on all 3. This is sort of a strong intuitive design. I feel better, my radiology is better, and my serology is better. That sounds good. The flaw is that the serology part of the EORTC score is something that improves very slowly, and that can actually cause that score to not work very well. What you really need to do with the EORTC MSG is focus on clinical response. There have been proposals to use point-based schemes. The MSG scoring system that's been used in many studies is basically point-based. The FLEET study added symptoms and fever and hypoxia to that. The difficulty with point-based stuff and percent point reduction is that it assumes points are additive. Does obtunded 3 points really equal a comp fixed titer of 1 to 16, which is also 3 points? It's harder for me to understand. Patient-reported outcome tools are sort of the holy grail these days for doing clinical trials. No PRO exists for COXI. The FDA released a request for companies to work on this project, and actually F2G applied for and received that grant. And so we're currently developing a PRO that's meant to capture relevant aspects of how a patient feels and functions. And some of the early work on that was presented recently at the mycosis study group meeting. We also have to solve for duration. When do you measure the endpoint? At least with azoles, this is a slow disease. Intervals of 4 months have been used in multiple studies. And if you want to do relapse, it's going to take a long time. And then when you're starting to do the statistics, you need to know the effect size with those endpoints at that duration. How big of a change does the azole get? How big of a change do you think your drug would get? How big does the study have to be? You got to have all that stuff before you can do the statistical part of the design. And then just like for any given new drug, how do you develop phase 2 dose finding data to inform phase 3? And we don't argue for a while about what comparator to use in the RCT. So, for me, I think it's surprisingly hard to design an RCT in COXI. You can spend a little time on the question of does it have to be an RCT? And John Galciani, I'm thinking about you here. Rob Hemmings gave a great talk at Trends in Medical Mycology last year, where he talked about times when you don't need to do an RCT. And there are a limited number of such settings. The classic examples would be some of those childhood diseases where you've got like spinal muscular atrophy. Every kid with that fails to walk at a certain time point. If somebody turns around, you actually don't need a whole lot more data to understand what that means. Other reasons not to do an RCT, well, there aren't really very many good ones, to tell you the truth. So when you're not doing an RCT, you're really probably doing a single-arm trial, and there are a couple of ways to approach the idea of a single-arm trial. There's the idea of just looking at outcomes in a disease that never, ever, ever, ever gets better on its own. And I think you can argue that, endocarditis doesn't cure itself, and extrapulmonary COXI doesn't seem to cure itself very often. I think it's an interesting idea. And you just say, look, I would not expect this response. You can also include some external control data and attempt to do some kind of a matching activity. And those approaches are mutually complementary. All those approaches are biased, but it doesn't make the data uninterpretable. You see a really large effect that's obviously just completely unexpected. That's very interesting. Okay, so in summary, this slide. Developing a new drug for COXI is harder than I would have thought. The basic requirements are substantial. It needs to be oral. You got to have a lot of safety data. You're going to have to have a lot of drug supply. Clinical requirements are very substantial. There are no easy trial populations for an RCT. This makes phase 2 dose finding hard. There are no standard comparators, and the available endpoint tools are limited, as are performance data on those tools that you would need to design a study. And I'll close with this slide, which is I think that we need to talk about what is adequate for new drugs for invasive fungal infections. And I don't want to use the word regulatory hurdle except one time, which is to tell you don't use that phrase, because the regulators just articulate the thing that everybody wants to know. We want better data. And when you hear we need an RCT, I want you to think about feasibility and what you can learn about other kinds of data, what you can learn from natural history. There's no easy fix for the challenge of rare pathogens such as COXI. Just point out one other thing about RCTs. They only study a limited subset of patients with a disease of interest. People sometimes don't get into RCTs. Some of the most interesting patients don't get into RCTs. And superiority, the idea that RCTs are superior design RCTs are smaller isn't always true. And I think we as a community have to engage on this. The rare nature of fungal infections makes this a critical conversation. And the core question can be stated a lot of ways, but really boils down to what's enough data to establish benefit risk for a rare orphan drug frequency infection? How far do you want to stretch statistical boundaries? And with that, thank you very much for listening. I appreciate the opportunity again to participate in this conference from a distance. I wish I could have been there. And I think we're now going to flip over and do questions.